Photosynthesis Lab by Bailey, Kylie, and Sam.
Photosynthesis equation is 6CO2 plus 6H2O. Then after light energy enters, it is C6H12O6 plus O2. The plant gets carbon dioxide from the atmosphere through small holes on the bottom of the leaves called the stomata. When plants get their water from rainfall, it is absorbed through their roots. The water is then transported to the leaves via the vein.
Our hypothesis was if we use a higher power lamp, then the rate of photosynthesis will increase at a faster rate because there will be more energy. Our outcome was that our hypothesis was incorrect because the light we used did not give off enough energy for the chads to float faster. They floated at a slower rate in the experimental group than in the control group.
Our procedure was to first punch 12 leaf discs with a hole puncher. Next, we put 10 of the discs into a syringe. Next, we pushed a small volume of air in the syringe. We then drew a small volume of sodium bicarbonate solution and inverted the syringe and tapped it to suspend the discs in the solution. The next step involved pushing as much air as possible out of the syringe. We then created a vacuum by holding a finger over the syringe opening. Once we released the vacuum, the discs sunk. We then poured 10 discs and solution into a cup and placed it under the lamp. We put a small amount of water in the petri dish and placed the dish on top of the cup with the discs in it. Finally, we turned on the lamp and started timing. We recorded how many leaves floated to the top each minute for a total of 20 minutes.
The materials that we used for this experiment were 10 discs hole punched from the spinach leaves, 200 milliliters of the 2% sodium bicarbonate, 1 syringe, 1 glass beaker, 1 petri dish, tweezers, an incandescent lamp for our original experiment, a high-powered lamp for our variable on the second experiment, and a timer.
In our control group, we used an incandescent lamp causing a fast rate of photosynthesis.
In this experiment, we used a more high-power lamp producing more light. This light caused the leaf discs to have less energy and float at a slower rate compared to the incandescent lamp. This change in sunlight caused a slower rate of photosynthesis. There was no change in the number of discs floating to the surface. Analysis discussion questions.
What is the purpose of creating a vacuum with the syringe? This was to remove all the oxygen from the leaf discs and cause them to sink into the sodium bicarbonate to the bottom so that once the sunlight was involved, photosynthesis can take place.
The leaf discs got their carbon dioxide from the sodium bicarbonate.
What made the leaf discs float? The sunlight made the leaf discs float, activating photosynthesis, producing glucose and oxygen. The oxygen in particular is what caused the discs to float. Once all the discs floated to the top, we saw that photosynthesis occurred.
Some of the sources of air within our group were possibly not getting all of the oxygen out of the syringe during the process, not suctioning the syringe correctly to make all of the chads sink, giving off too much carbon dioxide. When we hole punched the leaf, we could have caused a crease in the leaf, resulting in a slower or faster rate of change. When using the tweezers, we also could have punctured a hole in one of the leaf discs as well.
Compare experimental data with the control group data. Did the rate of photosynthesis change? Explain this result. What part of the photosynthesis equation did you alter, or did you alter the environmental factors? Our group changed the amount of sunlight in the experiment. The rate of change concluded in a slower reaction time of photosynthesis due to the change of the sunlight. This can be an alter in the equation, but also a change in the environment.